Every medicine you trust today exists because medicine once failed humanity. Some experiments save millions, others change history for terrifying reasons. Tonight, we journey into the shadows to uncover the dark history of medicine. Before the rigorous regulations we have now, the world of medicine was like the Wild West. Drugs were sold without any real safety testing. There were no ethics committees to review experiments, no structured clinical trials, no laws to protect the patients who put their trust in doctors. It was a world of blind faith, and sometimes that faith was tragically misplaced. One event, one heartbreaking disaster, would force the world to wake up. This is the story of the 1937 sulfanilamide disaster. Sulfanilamide was a miracle drug, one of the first effective antibiotics. It saved countless lives from bacterial infections, but it came as a pill, which was difficult for children to swallow. A pharmaceutical company saw an opportunity and decided to create a liquid version. To dissolve the drug, their chief chemist used a chemical called diethylene glycol. You might recognize that name. It's a primary ingredient in antifreeze. It's a poison. But back then, no toxicity testing was required by law. The company bottled it, flavored it with raspberry, and shipped it across the country. The reports started trickling in, then flooding in. Patients, many of them children, were dying in agony from kidney failure. Doctors were helpless, watching their patients suffer after prescribing a medicine they believed would heal. Panic spread. The final death toll was over 100 people. Children, families, patients who simply trusted their doctor. For the first time, society was forced to confront a terrifying reality. Medicine, the very thing meant to save us, could kill us. The public outcry was deafening. This tragedy directly led to the 1938 Food, Drug, and Cosmetic Act in the United States, which mandated for the first time that new drugs must be proven safe before they could be sold. The first major brick in the wall of modern patient protection was laid, cemented by loss. Just 2 decades later, the world was about to learn another brutal lesson. In the 1950s, a new drug emerged from Germany, promoted as a wonder drug. It was called It was marketed as a completely safe sedative. So safe, it was recommended for pregnant women to help with morning sickness. Doctors across the globe prescribed it with confidence. It was sold in nearly 50 countries. Then, a horrifying pattern began to emerge. In hospitals around the world, an unprecedented number of babies were being born with severe deformities. Many had flipper-like limbs, a condition called Others had internal organ damage, or were born without ears or eyes. The connection was eventually made back to thalidomide. The scale of the tragedy was immense. An estimated 10,000 babies were affected worldwide. The drug that was supposed to bring comfort brought unimaginable suffering. The world learned a painful, unforgettable lesson. Safety must be proven, not just assumed. The thalidomide crisis was a global wake-up call, shocking governments into action. It led to much stricter drug testing and approval processes, specifically requiring that drugs be tested for their potential to harm a developing fetus. The idea that pregnant women were a vulnerable population that needed special protection became a cornerstone of drug development. We learned that the promise of safety is not enough. It must be backed by rigorous, undeniable evidence. But what happens when the failure isn't a chemical, but the ethics of the researchers themselves? For this, we must turn to one of the most infamous episodes in medical history, the Tuskegee syphilis study. Beginning in 1932, the U.S. Public Health Service enrolled 600 African-American men from rural Alabama, 399 of whom had syphilis, into a study. They were told they were receiving free medical care for bad blood, a vague local term for various ailments. In reality, the study's purpose was to observe the natural, untreated progression of syphilis. For 40 years, these men were deceived. They were not given proper treatment. Even after 1947, when penicillin became the standard effective cure for syphilis, researchers actively prevented them from seeking treatment elsewhere. The men suffered. They went blind, developed mental health problems, and experienced severe health complications. They passed the disease to their wives, and some of their children were born with congenital syphilis. The study only ended in 1972 when a whistleblower leaked the story to the press, causing a firestorm of public outrage. The Tuskegee study exposed a complete and total failure of medical ethics. It led to the establishment of modern ethical guidelines, including the requirement for informed consent, where patients must be fully told about the risks and benefits of a study before they agree to participate. It also led to the creation of institutional review boards, or IRBs, which are committees that must approve and oversee all human research to protect participants. Our modern research ethics were written in response to this profound human suffering. Even with better regulations, accidents can still happen, and the consequences can be devastating. In 1955, America was celebrating. Dr. Jonas Salk had developed a polio vaccine, a beacon of hope against a disease that crippled thousands of children each year. A massive vaccination campaign began. But at one company, Cutter Laboratories, a manufacturing failure allowed live, active poliovirus to get into batches of the vaccine that were supposed to be inactive. Before the mistake was caught, about 200,000 children received a vaccine containing the live virus. Around 40,000 developed a mild form of polio. Over 200 were left with paralysis, and 10 children died. It was a national disaster. An attempt to protect children had instead infected them. The incident nearly shattered public trust in vaccines entirely. But science and public health didn't give up. Instead, they responded with reform. The event led to the creation of a much more robust system for vaccine oversight, with federal surveillance and stricter manufacturing standards to ensure the safety and potency of every single dose. The Cutter incident taught us that even with a miraculous discovery, execution and vigilance are everything. Fast forward to the turn of the new millennium. We enter an era of modern blockbuster drugs, where a single pill can generate billions in revenue. Vioxx was one of them. Launched in 1999, it was a new type of painkiller, a COX-2 inhibitor. That was supposed to be gentler on the stomach than older drugs like ibuprofen. Millions of people with arthritis took it daily. It was a massive success, but then troubling data began to surface. Studies started linking Vioxx to an increased risk of heart attack and stroke. For years, the debate raged in the scientific community. Finally, in 2004, the evidence became undeniable. The drug's manufacturer Merck withdrew Vioxx from the global market. It was a modern medicine wake-up call. Vioxx had been approved by regulators around the world, but this crisis proved that approval isn't the end of the safety story. It's just the beginning. It highlighted the critical importance of post-market surveillance, or pharmacovigilance, ongoing monitoring of drugs after they are released to the public. This event strengthened the systems we have today to continuously track the safety of medicines, ensuring that we can spot and react to risks that only become apparent after millions of people have used a drug. It was a painful reminder that even in our advanced age, we must remain ever. The quest for medical breakthroughs always carries risk, especially when we venture into truly uncharted territory like the human genome. In 1999, an 18-year-old volunteer named Jesse Gelsinger joined a gene therapy trial at the University of Pennsylvania. He had a rare metabolic disorder, but it was managed with diet and medication. He wanted to help researchers find a cure, especially for babies born with a severe form of the disease. The therapy involved using a modified virus to deliver a correct copy of the faulty gene into his liver cells. But just for days after receiving the injection, Jesse suffered a massive, unexpected immune reaction. His organs failed, and he passed away. His death sent shockwaves through the scientific world. He was the first person confirmed to have died as a direct result of gene therapy. All gene therapy research was put on hold worldwide. It was a moment of deep reflection for the entire field. Scientists had to go back to the drawing board to understand what went wrong and how to prevent it from ever happening again. The pause, born from tragedy, led to the development of much safer viral vectors and more cautious clinical trial designs. Today, gene therapy is no longer a distant dream. It is a reality, curing diseases once thought impossible, like certain types of blindness and bubble boy disease. That progress was made possible by the hard lessons learned from Jesse Gelsinger's sacrifice. Even in our hyper-modern healthcare system, with all its checks and balances, dangers can still lurk in unexpected places. In 2012, a mysterious outbreak of fungal meningitis began sweeping across the United States. Patients were showing up with severe symptoms, and doctors were baffled. The source was eventually traced back to a single place, a compounding pharmacy in New England. Compounding pharmacies mix custom medications for specific patient needs. In this case, They had produced steroid injections for back pain, but due to unsanitary conditions, those injections were contaminated with a deadly fungus. The contaminated medicine was shipped to clinics in 23 states. Over 750 people were infected, and more than 60 died. It was a horrifying revelation. These were not experimental drugs, but routine medications, prepared in a facility that was supposed to be safe. The disaster exposed a major regulatory gap. Compounding pharmacies operated in a gray area, not fully regulated like large drug manufacturers. The tragedy led to new laws giving federal agencies more oversight over these facilities. It taught us that medicine is only as safe as the weakest link in the chain, and that vigilance is required at every single step, from the giant factory to the small local pharmacy. So, why have we walked through this dark history? Because every tragedy we've discussed built the foundation of modern medicine. The sulfanilamide disaster gave us pre-market safety testing. The thalidomide crisis gave us rigorous trials for birth defects. The Tuskegee study gave us informed consent and ethical review boards. The Cutter incident built our modern vaccine safety system. Vioxx strengthened post-market drug monitoring. Hissy-Gelsinger's death made gene therapy safer. And the compounding pharmacy outbreak reinforced the need for supply chain vigilance, clinical trials, ethics approvals, drug monitoring, manufacturing regulations, patient protection laws. These aren't just bureaucracy, they are layers of protection, each one built on a lesson written in human suffering. Modern medicine is not perfect, but its greatest strength is that it learns. Every safe drug you take, every vaccine you receive, every ethical trial conducted today carries the legacy of those who were harmed in the past. The dark history of medicine is not a story of failure. It is the story of progress. It is the story of how, time and again, humanity has been confronted with its mistakes and chosen to do better. Thank you for joining us on this journey. If you find these stories of medical history important, make sure to subscribe and hit that notification bell for more. See you next time.